Welcome to MDLT 125, Phlebotomy Laboratory Techniques, Lab 9, Laboratory Equipment, Microscopy, Wet Preparation. So the theory, some samples can be placed directly under the microscope. However, many samples look better when placed in a drop of water on the microscope slide. This is known as a wet mount. The water helps support the sample and it fills the space between the cover slip and the slide, allowing light to pass easily through the slide, the sample, and the cover slip. We will now go to the practical aspect of this. Okay, class. So now we're going to do the practical aspect. So we're going to make the wet preparation. We would need a sterile cotton tip applicator. So where it says to peel, we're going to peel to open. Right, so you can now take a sample from whether it be around, whether the desk, amongst yourselves, but of course we're not sharing the swab amongst ourselves, right? One per person. So we're going to take the sample, and after we've collected the sample, we place it in saline solution. We'll take a microscope slide. Take your swab, take out the excess, place it onto your slide. And we're going to add the water. But before we add the water, we have to remember that there's a technique in order to use the pipette. You should place the pipette between your fingers, whichever one is comfortable, this way, this way. You will squeeze on the bulb, go into the solution, and then release. Afterwards, you place one drop then the next thing is to add the cover slip. You must bring the cover slip as close as you can onto the slide to prevent bubbles. And that's it. So we're continuing with Lab 9. And with this part, we're going to be doing cell counting. So cell counting, we need to use an instrument called the Improved Nubar counting chamber. So it's also known as the hemocytometer or hemocytometer. So this is what we're going to be using to count cells, and this is specifically done in hematology for red blood cells, RBCs, white blood cells, WBCs, platelets, and it also can be used in biochemistry, well, according to which department you are in the laboratory, you can do the semen analysis using this instrument. So the one earlier was procedure 1, so we're moving on to procedure 2. So procedure 2 entails the hemocytometer or the hemocytometer, which is this device. So what you will observe, first observation would be if you compare it to a slide, it has the same dimensions as a slide. But the thing about it is not quite a slide, wherein we can replace a slide with just, don't want to say cents, dollars. This will cost thousands of dollars. So we don't want to break this instrument, even though it looks like reasonable to replace. So the name of this is an improved Neubauer, right? And what you notice, if you can notice, is there is a upper and lower part of the actual counting chamber. So when you use it, you're able to read from the upper and read from the lower. And how you would know that your technique is done well is the number of cells that you will count on the upper to the number of cells that you count on the lower will be similar. There will be not a great disparity between the amount of cells that you count. So each, let's say you're going to count white blood cells. You have specific diluting fluids for each one of them. So you cannot say you're going to count RBCs, red blood cells, and you're going to use WBC counting fluid. You will not see anything, or vice versa. The other thing that is required to use this instrument is a cover slip, wherein we also use a cover slip in the wet mount or the wet preparation. This cover slip is not disposable. So you should not throw this. This can be reused. I don't know if you can actually see that it is thicker by far than a normal cover slip, and the actual dimensions is made to fit over the area which you're going to be using on the counting chamber. So I'll do that now. And then you would now take your preparation of cells with diluting fluid, and you will now insert in this area and this area.